If you haven't met me yet, I'm Paul Frost, and I co-organized this workshop with Angie. She did most of the heavy lifting, and I sort of just drifted along and provided moral support mostly. That's my story. And we started talking about this as she mentioned at the conference on biological stoichiometry COBS a couple years ago. And I was interested in the whole topic ecological stoichiometry and toxicology because I've been thinking about it for a while. Some of this goes back into the early 2000s, and it started with actually James Larson, so he's here, which is nice. I can't say that this long period of incubation has led to much, though. So you're going to sort of see really immature thoughts. We've been thinking about it a long time, but we haven't really thought very hard about it. So we have sort of the worlds of stoichiometry, toxicology, and math crashing together. And the idea for these talks was to sort of introduce each of the areas to people who might not be familiar with them. So we saw the toxicology earlier this morning, and I was given the task to talk about ecological stoichiometry. And I know some of you are very knowledgeable about ecological stoichiometry, so sort of just go with me on this. For those who aren't familiar, we're going to start kind of from the basics and build up. And the first thing I like to talk about with ecological stoichiometry is still a relatively new way of thinking. So if you go into Web of Science and you search for this key term, ecological stoichiometry, this is the distribution of papers that you see through time. So before 2000, this is not to say it wasn't happening, but people weren't really using this term. And you can see there's been sustained and continued growth in this field over the last 15 or 20 years. So 727 papers now published with this key term. And in the last 3 years, this is about 70 to 90 papers, so a substantial number of papers per year. When I started back here, you could read a paper a month or so and sort of keep up with what was going on. Now, good luck. You know, you're not going to, that's a lot of papers to keep track of. And this has had a significant influence on ecology. So I like to argue that this isn't just some sort of insignificant field, because if you were to take the total citations of these 700 papers, that's 20,000 citations they've generated. You could be a little more conservative and get rid of self-citations and just drop down to around 16,000. Still a lot of ecological influence this is having. The other thing is I find interesting is if you take this number and divide by that, that's 27 citations per paper. And if you think about those papers not actually being cited much yet, so there's actually a pretty significant influence of each of these papers on ecology. It's grown so much that now you're actually seeing stoichiometric publications. This is the stoichiometry newsletter. If this is my chance to sort of plug this. So it's called Ratios Matter. This has been going for a year. This is the website. If you're interested in having a free subscription to Ratios Matter, you can talk to me later. Or you can go on the website and fill out a subscription, free subscription. You guys are laughing.
Oh, it's free? Do you plan on charging?
We have a growth model. This could be in the future. At this point, it's completely free. And the other thing about that is that we are really interested in stoichiometry stories. So if you work in ecological stoichiometry and you have something you want to share, this is a great outlet because we have pretty low standards, right? There's almost no bar to get into this. It could be a picture, it could be a graph, it could be anything. So this is an outlet, and we have like, I don't know, 500 or 600 subscribers. So if you wanted to share something with people who like stoichiometry, that's not too bad to have this sort of like thrown into their desk and they would see what you have to say. So go check it out. More famous stoichiometry text is this book, which was written by Sterner and Elser in 2002. And I just bring this up because this has sort of been the book that started a lot of this. This has got a lot of people thinking about it. It's been cited a lot. And it provides a nice definition: the study of the balance of chemical elements in ecological interactions and processes. So that's what we're going to talk about, ecological stoichiometry. Now, there's different ways sort of think about ecological stoichiometry and what that means. So I'll talk about the way that I think about it and how I deal with this. It's not to say that there aren't these other perspectives or this is the full way to think about it. But this is where I start. I start at the consumer-food interaction. So to me, this is a key part of ecological stoichiometry, and this is an example. Here you have Acellus, which is the little pill bugs. I don't know if you've ever seen these. A lot of times they live like on your driveway under a rock or something, but there's also cousins of these pill bugs that live in streams, and they eat this stuff, which is this leaf material. So you go to a stream, you take the net, you dig around in that leaf material, and then you dump it out into a bucket, and you'll find these Acellus, and they're really cute crustaceans. They parthenogenetically reproduce, so they're kind of like a Daphnia analog, except they're kind of hokey pokey. And they tend to have nitrogen-rich material in their bodies like a lot of animals, and they eat this material that has nitrogen poor, which is a nitrogen poor food. Nope. And so this creates what I call an elemental mismatch. So the nutrients in their bodies don't match that they're consuming. So this elemental mismatch is really what stoichiometry is dealing with. What are the causes and consequences of that? So there's a couple ways to think about that. The causes part is what controls the nutrient ratios in the food material versus that in the consumers. So if you were asking stoichiometric questions about that, you might talk about nutrient supply, you might talk about light and nutrient availability to primary producers, you might talk about the balance of nutrients flexing through the environment, and you'd have to couple that to the physiology of the plants and how they deal with those imbalances and what that means for this material. Now, in this case, Acellus is being exposed to residual plant material, so you would then have to sort of add in another filter, which is like, what did the plants do before they got rid of these leaves that fell into the streams? And the other part of that is what controls the nutrients in the bugs. So there's a whole field about where are these nutrients at, and how does the biochemistry drive into this, and how does this connect to life history? And so those are all kinds of questions that are bound up in this. The other part that stoichiometry has spent a lot of time thinking about is what are the ecological effects of elemental mismatches. So an Acellus eats a nutrient-poor food. They might grow slower. They might change their nutrient release. They might have different reproductive rates. And then these can, if they're multiplied across the population, have community interactions, change competitive interactions, stuff like that. So those are all great questions, and it's sort of, we could spend more or less a whole day sort of going through how those all work. We're going to focus and just think more about this physiological part just for a while, because this is sort of the stuff that I've thought most about. I think this is still at the heart of stoichiometry. I call this physiological stoichiometry, sort of like what's the effects of imbalanced nutrients on the physiological processes in organisms. And we thought about this in sort of three different areas. So if you're a consumer, and it's represented by this, I guess that's a cylinder. It's an ugly looking consumer. And they have a certain combination of carbon and phosphorus that they need to grow. And then they're eating this food. Giant food particle goes through them. And then what they don't incorporate is released as waste. So here you can see in this example, there's 3 C's here and 3 P's. The consumer keeps 1 C and 2 P's, and that would yield 2 C's and 1 P release. So this is sort of a mass balance part. Stoichiometry is controlled by a mass balance. You can't make carbons up. You can't destroy the phosphoruses. What you consume either goes into the consumer or is released as waste. That's a constraint of the system. There's really no way to get around that. So if a consumer eats a food that has different combinations of elements than what it needs, it can do some things. So this can change feeding rates, food selectivity, digestive and absorptive efficiency. So these are all mechanisms that consumers can use to sort of balance out these differences in the elements being supplied versus what they need. They're not entirely successful at fixing this. So even though they do all those things, consumers still feel effects of nutrient imbalances on their growth, reproduction, and elemental storage. So these sort of like the physiological consequences of eating nutrient-poor food. And then finally, as consumers are trying to fix this difference between what's in the food and what's going in their body, you start seeing changes in the ratio of release. Some of these are used by the consumer to try to balance the imbalance. So like respiration rates tend to increase if a consumer eats nutrient-poor food. It's because they're trying to burn off excess carbon. And that's sort of the prevailing stoichiometric thought. So you sort of have changes in acquisition, incorporation, and release if you have differences in the nutrient ratios in the food versus that in the consumer. So this, as I mentioned, can result in skewed nutrient release ratio. So a 1-to-1 ratio, this is the way, as a stoichiometrist, you might actually show this, is you would calculate the ratio of C to P. So here's a 1 C to P ratio, but the consumer needs half. And if there was a difference in the consumer body versus the food, then this would result in a skewed nutrient release ratio. We could play around these. The food doesn't have a C to P ratio of 1, just in case you're curious. It's about 100 to 1. And consumers have a C to P ratio of about 100 to 1, but the food can go up to 1,000. So this creates the possibility that there's these big differences. This was originally recognized. This is sort of the start of modern stoichiometry. So I'm going to show you sort of the origins of stoichiometric thinking, and it was really through that skewed nutrient ratio idea. And so this was one of the earliest models of stoichiometry, and what they were thinking was the N to P release ratio was a function of that ingested minus that incorporated divided by phosphorus ingested minus phosphorus incorporated. And you can specify ingestion rates and incorporation rates if you know how fast the organism is eating, the food nitrogen phosphorus ratio, assimilation efficiencies, and what the animal. So you kind of have to have some information on how much elements are being delivered, how much are being kept, and then you can calculate what's being released. So in one of the earliest, more or less explicitly stoichiometry papers, Bob Sterner did this back in 1990, and this is a paper from American Naturalists, where used mathematical formulas to sort of place constraints and place limits on the movement of nitrogen phosphorus through consumers. So what's really interesting about this and what's shocking is if you go to an algae that has a fixed N to P ratio, this is about 15, depending on the consumers, different consumers are denoted by the different lines. If you had a consumer with a high N to P ratio, it would release an N to P ratio that's really low. On that same food, if you fed it to an animal with a low N to P ratio, it would in theory release an N to P ratio that's very high. So you could have really different nitrogen and phosphorus fluxing from the consumers back into the dissolved pool. You could change this dramatically even though you haven't changed the food at all. And this is just simply reflected by the N to P ratio in the animals. So by increasing the nitrogen to phosphorus ratio in the animals, these animals have more of a phosphorus need, so they're holding on more phosphorus and releasing more nitrogen. This releases a high N to P ratio. These animals have a low phosphorus need, so they're holding on more nitrogen and releasing more phosphorus. So this is inherently stoichiometric, and it shows sort of this nonlinear and unexpected dynamic whereby changing the N to P ratios in the animals results in hugely different N to P ratios in going back into the dissolved pool. And the other thing is for an animal of a particular N to P ratio, changing the N to P ratio in the algae results in a higher N to P ratio. So you make the algae more nitrogen rich, you end up with a higher N to P release ratio, but it doesn't fit this 1-to-1 line. So it doesn't sort of, it's not a perfect correlation. It creates this sort of non-linear relationship. So this hinted at some sort of more complicated dynamics or some more interesting phenomenon going on here that was discovered by knowing the animal nitrogen to phosphorus ratio, and that there was stoichiometric mechanisms at play. So that got people, like a lot of early stoichiometry was dealing with nutrient release ratios. It was fascinating that you could change the N to P ratio going back to the dissolved pool. But it was also noted that if you fed an animal low phosphorus food, they would grow slower. So this is one of the most repeated experiments in stoichiometry. I think probably the most amount of data that shows that if you feed animals a high carbon to nutrient ratio food, they're going to grow slower. So here I've changed the numbers around just to sort of show why that is. If an animal is eating food with a C to P ratio of 3 to 1, but it needs a C to P ratio of 0.5, it basically has to eat 2 of these to get enough phosphorus. And so now it's eating 6 C's and 2 P's so that it can get the 2 P's so it can grow. But it ends up having so much extra carbon that it has to then get rid of a lot of carbon. So in this case, it's perfectly efficient. It keeps all of the phosphorus in the food, but it has to release this crap load of carbon. So again, this goes back to this is enormously skewed release ratio because it's not releasing P. But it hints that the rate that it can actually incorporate carbon and phosphorus in this ratio will be constrained by the rate that it's being delivered by it eating this food. So if you reduce the P content in this food by stuffing more carbon in here, they're going to have to eat more and more food just to meet the delivery rate of the element that's limiting. And in this case, we're assuming that it's not the carbon, right? You sort of get into this realm where you're limiting growth not by the quantity of food. You can eat as much as you want. You're limiting growth by the element in least supply. So a lot of stoichiometry has been built on this, sort of trying to understand under what circumstances you would be limited by this element in least supply beyond carbon. And you can do these experiments. So this is sort of where I come in. This is sort of sets up the stage where I say, okay, well, let's do a bunch of experiments with Daphnia and let's see how these systems behave. So this is a sort of typical Daphnia system, stoichiometry of Daphnia. You have a single food source here, which is Synodesmus, is the type of algae. If you change the phosphorus supply to Synodesmus, you can change the carbon to phosphorus ratio in its cells from 100 to 1,000. If you flip this over and calculate the phosphorus to carbon ratio, it makes these inverse, and what you see is this is about a 10-fold change in the phosphorus content of the algae. The Daphnia have a body carbon to phosphorus ratio of about 100. Now this can change. You can talk to us about how much it changes. It doesn't change anywhere close to this. The highest C to P ratio we've measured in Daphnia in our lab depends a bit on the species. Probably the highest you'll ever see is something like 150. That would be really high. Typically, a phosphorus-limited Daphnia would have maybe like 110, 115. A really phosphorus-rich Daphnia, this ratio would actually be somewhere like 70 or 80. So you're really talking about a different range in C to P ratios in the body than what's seen in the algae. So here you have the Daphnia in your lab. Oops. You feed them some food. So we have different food CP ratios. This is Daphnia magna, and this is the growth rates. You can see feed them a higher CP ratio, and they grow slower. And you do this with different species. You get slightly different slopes. You can, well, the other question is what is relevant? So you can make an algae a CP ratio of 2,000, but in most lakes, the C to P ratios sit around 200. And there's many lakes where CP ratios are 100. You can find it on a reasonable basis, depending on where you're at, sometimes being as high as 400 or 500. So usually we do these experiments sort of like the 100 to 600 range or something like that. So if we create this elemental imbalance at the Daphnia food interface, it not only affects their growth rates, but their reproduction. So you can do these kinds of experiments. So here's the CP ratio of this Daphnia, again, around 80 to 100. And here we change the food CP ratio going up to 1,200. And in this case, we kept the Daphnia in the lab for 30 days. And this is a lot more work because you have to figure out the phosphorus content of the food and then make the right mixtures. And then you're changing the media every couple days. And then after around 7 days, the Daphnia start reproducing. So you have to go in the lab every day and remove the neonates. So it's pretty intensive. You don't really want to do many of these 30-day experiments. My students keep wanting to do those, and I say, I don't know why you want to do that. But it's really nice what it gives you because it allows you to calculate reproductive measures in survival. So in this case, we just counted the number of broods. What you can see is there's a huge drop-off in reproductive events over 30 days if you're feeding Daphnia low phosphorus food. So not only are they growing slower, but now they're not reproducing as much. And part of that is because the day of first reproduction gets extended out. A phosphorus-limited Daphnia reproduces not on the 7th day, but can be as delayed up to like the 20th day. So that's pretty bad for them. And at one point, we were curious what these offspring would look like. So if you took neonates from these animals, would they be the same as those? And no, not really. So this was what we called transgenerational effects. So these are neonates from the same Daphnia clone, but Daphnia moms who are eating different foods. So these moms were eating phosphorus-rich food, phosphorus medium, and phosphorus-poor food, and then these were the neonates that came from them. And you can see there's a big difference in their size. And what's interesting is we then took these offspring and we fed them food at different food qualities, so this sort of gets a little bit mind-bending. But on the same food quality, they do different things than what they do. So this sort of stoichiometry changes between generations. And the reason for this is because coming from a phosphorus-poor mom, they're provisioned with less phosphorus. So when they're presented then phosphorus-poor food, they do far worse than these guys do. So the stoichiometric effect sort of gets amplified the second generation. But the other interesting thing we found, and I'm still a little suspicious of it, but I think it's legit, is that when these were presented phosphorus-rich food, they actually grew about 10 to 15% faster than these guys. So they were sort of like trying to make up this deficit. And we suspect that they were trading off this fast growth for some other loss in function. We're not quite sure what that is. So if you feed the Daphnia phosphorus-poor food, they grow slower, they reproduce less, they start producing different kinds of offspring. It also affects their survival. This is data that was just taken from my master's student who just finished, and he fed the Daphnia different food qualities, CDP ratio, so phosphorus-rich food to phosphorus-poor food, and then half of them he exposed to predator cues and half he didn't. So that's why you see fish cue with and without. And I'm not really talking about the cues, but this is kind of interesting. I just wanted to show you what happens to the Daphnia over 30 days in terms of the proportion living. So if you're eating a phosphorus-poor food, at the end of 30 days, you're somewhere like 25 to 50% survival. Whereas if you're eating phosphorus-rich food, fine, no problem, 100% survival. So this starts really changing the demography, right? Like starts changing their potential population growth, especially if you couple that to this. So not only are you not producing offspring, but you have a much higher rate of mortality. So bottom line here, elemental imbalances are bad for consumers because they alter life history traits underlying changes in population dynamics. You can really change these population trajectories by changing food quality. And I think we know that. So, but this is an illustration of stoichiometry and sort of like it's how elemental imbalances affect consumers. And it can change all those kinds of things. So here we have stoichiometry, but what about toxicology? We're going to try to see what this means for toxicology. Well, it's maybe not surprising because toxicology deals with a lot of stuff that I was just talking about, right? Growth rates, reproduction. Would those things change if you change the ratio of nutrients available to a consumer? So this is kind of where I've been thinking about eco-stoichiometry toxicology interfaces for quite a while. Is at the consumers, if they're nutrient stressed, would they change? Would that change their responses to toxicology? So I hope there's no hardcore toxicologists here because I'm not very enamored with this view of toxicology. So we'll talk somewhat poorly about it for a minute. No disrespect, really. This is sort of the classic toxicology approach. You change the concentration, you see what percentage of your population of organisms is responding, so whether this is death rates or something like that. And then you use that to calculate this is an effective concentration or a lethal concentration. And generally, so this is the standard toxicology test. Generally, you'd want to do this under an optimal condition. Just like Ryan was saying, if you grew them in an unoptimal condition, this line would be kind of flat and it wouldn't really tell you about the toxicology. So you want at low concentrations, so sort of control, you want the animals to grow fast, you want them to all survive. And that allows you to see the level of toxicity as you increase the concentration. The problem with that, there's nothing wrong with that. So if you want to compare the toxicity of chemicals, if you want to compare toxicities between organisms, that's fine. The problem is when you say, we're going to use that information to sort of understand what's going on in the environment. Because the toxicity of the chemical might change depending on the physiological state of the organism, the environmental conditions, and that kind of stuff. So this seems to me like a relevant question for stoichiometry. How much would this response curve change with changes in elemental food quality? So imagine this is a Daphnia, and you're exposing it to a range of chemical concentrations. Would this line change if they were eating different foods? So we've thought about this sort of in a theoretical sense. So I'm going to sort of tell you the Daphnia story. What affects the toxicity of Daphnia? This is our hypothesis, elemental food quality. And we kind of have this starting point prediction, which is greater toxicity should be found in nutrient-stressed animals because constraints on the physiological repair mechanisms. I think this is pretty standard. I don't think you'll find a lot of disagreement that this seems like a good starting point. If you're not eating enough phosphorus or nitrogen, it's creating all these physiological responses, showing slower growth rates, less reproduction. It would seem like you would be less capable of dealing with stress from another stressor like toxins. So the general approach to do this is not real data, so just follow me here for a second. General approach would be something like this. Change the chemical concentration of some chemical, and here's the survival probability, and we have Daphnia eating phosphorus-poor to phosphorus-rich food, and you might see curves like this. So my learning curve being a toxicology person was, well, how do you know if you see a change there? And one way you can do it if you're sort of a real classic toxicologist is to use this data to calculate LC50. But the problem with doing that is that the LC50 formulations take into account the slope and the intercepts, and so it sort of couples two forms of uncertainty, and it yields a wider, correct me if I'm wrong, Brian, that's fair, isn't it? Yeah. And it sort of overly focuses on this. So actually talking with James about this a long time ago, we came up with sort of a log survival approach. This sort of pushed us out of mainstream toxicology, but in some ways it was more useful for us because we just wanted to know whether the slope of these lines would change. So the odds of survival equal the toxin concentration times some slope. So that would be the slope of this. And we just wanted to know whether that slope would be different, depending on whether the food quality was different. And because we then have these standardized slopes, we could estimate uncertainty associated with it. Again, not real data yet. And the other nice thing is you can run these experiments for many days, and then you can sort of show how these slopes change through time and how stable this is. And this starts getting at sort of like different stages or ages of the animal, stuff like that. So this would be, again, an example, different beta slopes. And if you look here, a steeper slope means you have a lower survivability with a given concentration. So more negative is more toxic. So negative number, more toxic. In this case, the theoretical animal eating the phosphorus-poor food was more affected by the toxin. So I'm showing you all of this because we did a bunch of these experiments. I think originally James and I wanted to sort of like go through this big book of all these chemicals and do screening tests. And then we sort of got overwhelmed with the chemistry between the potential toxin and the media that we were using. It kind of got really complicated, so we didn't maybe make as much progress as we wanted. And I'll just tell you a quick story. Again, pretty steep learning curve. I had an undergrad who was going to use copper sulfate. She was going to study whether Daphnia's responses to copper sulfate change with food quality. That's really great. Great stoichiometry, toxicology interaction. We found the toxicology literature. Here's the copper sulfate, Daphnia magna LC50. We're using Daphne magna. So this tells us about the concentration that where we would see 50% of the animals. Now, we weren't expecting to sort of like get that perfectly. She did a few experiments, I would say 3 or 4, and she was exposing them to 1 milligram of copper per liter, and none were dying. What? And then we realized that we had the Daphnia growing in an artificial growth media full of EDTA, and this was probably binding the copper. So for us to become toxicologists, we also had to be a little bit smarter chemists. So then we had to start growing them in natural river water, which sort of allowed us to do these experiments. I know I'm not instilling a lot of confidence in our results at this point, but it is actually kind of interesting what she found. So here's copper concentrations, and this is around that range. It's supposed to be the LC50s. And here we have phosphorus poor food. So you can see at no copper concentrations, we've made the Daphnia grow slower by reducing the phosphorus in their food. So this is pretty exciting, but not surprising. And then we increased the copper concentrations. There was actually a slight decline, but we didn't really see anything. That's not very cool. Then we did that log survival analysis that I was showing. Again, if you have a really steep slope, it's more toxicity. And here, it's weird because if you look at this, 1, 250, and 400, this is phosphorus rich to phosphorus medium. They all sort of, there's no change, but the animals eating the really phosphorus poor food showed less toxicity. Huh. So there was better survival in those animals when they were exposed to higher concentrations of copper. And this was more or less evident all the way through the experiment. So that was confusing. We'd sort of say, okay, well, let's keep going. So I had another in Canada, you get to have a lot of undergraduates who've cycled through your lab, and these are great experiments. You can kind of do like one a summer or something. So I had one student who was really interested in pesticides, and there was a group there at Trent that was working on Roundup and amphibians. So I said, well, why don't we talk to them? And unbeknownst to me, the way that we got our Roundup, I found this out later, is that grad student that we talked to went out, and she knew a farmer, and he just went out to his barn and got like a bottle of it for us. So this was actually the Roundup that they're using to put on their fields. And we did receive a little bit of criticism for it. The active ingredient is glyphosate, and the tricky thing they do with Roundup is they put different functional groups on it. And the toxicologists, again, if I'm off here, you can correct me. Toxicologists are trying to keep up with the company because they'll change these active additives, and they're sort of like always one step ahead of the regulators. And they formulate it differently so that it works differently depending on climate and soils and stuff like that. So it's kind of frustrating because most of the data is on glyphosate, but it really has to be specific to the formulation of the Roundup that they're coming up with. So there's a little bit of disconnect, but we use the actual stuff that they're putting on the field, so deal with it. So here we have glyphosate, and it takes a lot actually to kill a Daphnia in the absence of that extra stuff. Look what happens, and this is lower concentrations, but still a lot. Look what happens to the Daphnia growth rate. So at, again, control conditions, this is what you would expect. Daphnia eating phosphorus-rich food grow faster, and Daphnia eating phosphorus-poor food grow slower, and it's a nice linear relationship. And then as we increase the Roundup concentrations, at the higher concentrations, this is where we start seeing them drop off. This actually flips. So the ones eating the phosphorus-rich food are growing way slower than the ones eating phosphorus poor food. The ones eating phosphorus poor food kind of don't care, and the ones eating phosphorus rich food are completely hammered by this stuff. Strange. Again, it's not consistent with that sort of simple prediction that we put out at the beginning. A little less clear. I don't know why we didn't have other higher CP ratios, but probably because of the amount of work involved in keeping the animals alive for all of this. We see a similar sort of thing where the highest toxicity in terms of survival is for the phosphorus-rich food with the bottom line, and reducing phosphorus in the food increases survival in the presence of Roundup. So this sort of is, again, reduced toxicity in animals that are phosphorus stressed. So we're sort of like parade of chemicals here. We heard about the pharmaceuticals. This is a great one, fluoxetine. This is found in Prozac, and it's getting out into our wastewaters. Doesn't get removed by the sewage treatment plants. So there's a fair amount of work on this. This is actually one of the first instances where I worked on this, and the way I got into it was I had another undergrad who came to see me. This was the first undergrad I had, actually, and she said, "Ooh, I want to do toxicology." So I said, "Oh, why don't you go talk to the toxicologist down the hall, Chris Metcalf, who's measuring things." That's mostly what he does. And he said, "Oh, yeah, I have some fluoxetine in the fridge. You can use that." So we looked up its LC50, and here you go. I don't know who that character is. This looks like a great paper. So this gave us a starting point, 800 micrograms per liter for your Daphnia magna. So before I show you the results, though, I'll tell you it's a funny science story. So I was at home. We had this completely novice undergrad. So even below the honor student, he was like a second-year student. He was helping. And he called me, and he said, "There's something wrong with the experiment." I'm like, "What do you mean?" He's like, "All the ones in the phosphorus-rich media, they've died, but the ones eating phosphorus-poor food are still living." And he was like, "We did something wrong." I'm like, "That's not wrong. That's publishable." So I had to explain to him, like, sometimes in science these things happen, right? This is great. So so here we go. We changed the fluoxetine concentrations, and this is the growth rates. And again, at the control, high C/P ratios, phosphorus poor food, they grow slower, so that's really good. But when you get out to a higher fluoxetine, you can see animals eating the phosphorus-rich food start to grow slower, and the ones eating phosphorus poor food don't really care. And we saw this in survival as well. So here's the phosphorus poor food. They don't really change their survival, whereas there's a much steeper slope for the ones eating phosphorus-rich food. Now, the other thing we did with this, which was because we were interested in whether this was a phosphorus-only mechanism. So we also repeated this experiment once with nitrogen, and we saw almost the same thing with nitrogen. So we had to sort of conclude that this was not because the animals were phosphorus limited, but because they were nutrient limited. And this sort of brings up some discussion points about toxicity and growth rates and stuff like that. So if we put all that together, I think you can sort of make a preliminary conclusion that elemental food quality matters. A really nutrient-stressed animal processes food is differentially susceptible to toxins than a fast-growing, nutrient-rich animal. But we found that there's actually less toxicity in these three examples, which isn't consistent with that sort of simple hypothesis that nutrient stress increases toxicity. And I think this is something we need to talk more about, which is like, there's something about the physiology of fast growth, and I think it's on my next slide, but we need more of this. So unfortunately, the funding situations, and there aren't good programs, but if we can kind of keep chipping away at it, and I think if we had models that sort of informed us, like these are the kinds of chemicals we should work on, and these are the kinds of response variables, this isn't that hard to do. We can grow a lot of Daphnia, we can change the food quality. We need help with the tox part, we need help with the chemistry, and we need to sort of have some direction to measure, which response variables to measure. So I think the big question here is, what's the mechanism? Well, is it that when you're growing fast, you're processing more and you're just exposed more? That might be part of it. We need like a physiological toxicology person to help us with this. I actually think that something like this, like cell division, is especially sensitive. So if you're growing really fast, you're taking your DNA, you're making copies of your cells, and those division processes are really sensitive to exposure to toxins. And I suspect that a little bit of this is just that nutrient limitation slows this all down, and they're just not, either it's not as much exposure, or there's just less cell division, which is really sensitive. Sorry to sort of flip back. This one here, and I think this is the other danger that you sort of get into. This was the first time we did this, sort of like toxicology, stoichiometry with the fluoxetine. And I did a lot of hand waving during those years about Prozac and its mechanisms of interaction in the animals, and what's serotonin, like do Daphnia have, they don't have moods, right? I mean, we weren't in the lab trying to figure out if the Daphnia were depressed or anything like that. I suspected the fluoxetine was working on different mechanisms in the Daphnia than it would work in a human. So we have to sort of be careful when you jump organisms. One of the things I found, this is my hand waving part, we found serotonin's involved in embryogenesis. So it regulates cell division. And I just thought these guys aren't doing this, so they probably have less serotonin. But whether that is a general thing, I don't know. It's sort of, is that a chemical specific thing, or are there general patterns that sort of come out? Fast growth yields more sensitivity. I think is a good question that we need to sort of think about, and certainly how that changes between chemicals. So we're sort of at the end here. I don't know how much time I've taken. So I have sort of some general thoughts, and I'd be interested in answering questions. Chemical toxicity varies with nutrition. I think, that's pretty clear. The nature of the interactive effects could vary with toxin trait, nutritional components, and we need to sort of keep that in mind if we're creating more general mathematical models. And I think this is reflective of what we heard this morning as well. So this is sort of like a cautionary tale that we shouldn't we should be very careful about how we formulate the models and make sure that they're appropriately informed by the toxicology. And my interest here is models that capture sort of individual responses. I'd like to sort of work on that part of it anyway. That's sort of probably what I'll be doing. And so we're in with questions and discussion. Yeah.
Just a comment. Did you have a look how much of the compound actually enters your Daphnia? Oh, sorry. Right, so the question is whether you have checked how much of the compound actually gets into the Daphnia, so not the nominal concentrations, but the...
No. So this is a question I'm almost always asked. So we've done work with nanosilver along these lines as well. And so this is always sort of like, have you measured it in the Daphnia? And one part of my answer is for why we haven't, which I think I should answer that, is because, I don't know if we found that interesting, but the other part of it is sort of methodological. So animals that are 6 days old are pretty small, and the methods for measuring some of these things is pretty not routine. So like measuring fluoxetine, we'd be dealing with really low concentrations. So I'm pretty sure we could do it, but we'd have to sort of change the systems around. It's not the easiest thing to do, but it's an interesting question. I think in each case, we can be assured that we expose the animals to some gradient. I'm not sure we can be entirely sure we exposed them to the gradient that we say we did, because in some cases we use nominal values. This is where the toxicology people start chasing me out the room. But whether they're exposed internally and how much is accumulating, that's certainly a question that would be relevant. And I know, there's questions about growth dilution and stuff like that, which sort of come into that. It's a good question. So no, but certainly interesting.
So I have a sort of like a question. Can you go back to your last slide? the one with the data, yeah, this one, or before even, yeah, this one. Do you think there's a possibility, because this whole experiment lasts for about a few weeks, right?
So this experiment would have lasted 6 days. That's the standard Daphnia growth experiment. This one lasted 2 weeks.
Okay. So, to some sense, this resembles what we see that in sort of like, say, the chemotherapy, sort of like, or many of these sort of treatment for cancer or for sort of bacteria. So the resistance development, I mean, that's probably for the what sort of like have a lower growth rate, maybe stressful, maybe somehow leading to some kind of could be some physiology, sort of like a change, sort of allow the individual to develop a different sort of like growth pathways. And for the higher nutrient level, then of course this, I think I agree with your hypothesis that because there's more division, and then there's of course you are absorbing more sort of the toxicant into the cell, right? Because when you don't divide, the toxicant won't do anything to use. So maybe that sort of can be quantified to use to explain that.
Yeah, I think that would be the next step, would be to try to figure out the rate of delivery and then look for aspects of cell damage or something like that.
And how many individuals, typically the cohort, you have like, you are talking about hundreds of the Daphnia?
In this type of experiment?
Yeah.
I can tell you a little bit more about our typical methods. So like in a growth experiment where we would do this,
Right.
these are typically, I don't know, 5 or 10 animals
And then okay.
in a treatment combination. And the reason we don't do more than that is because we're using a clonal animal, so there's very little genetic variability in a standardized water with standardized amounts of food, so we don't see very much variation, right? So if we added 30 animals here, we would get the same answer. So this is just an efficiency issue. For this type of experiment, when you're growing them over periods of time, you're expecting some levels of death. So you sort of want to be able to sort of capture the mortality events over time. So in these kinds of experiments, you'd usually use trays of 25 Daphnia or even more. And then you would just keep those going for 30 days.
I see. So
Yeah.
but-
So it's a lot of Daphnia. I mean, in the big experiments we ran in the lab, I think at some point I had like 1,500 Daphnia. I don't really want to go back to those days, but you can do it. You can scale up. You can have really large, and these are all individual animals in individual tubes. You could also have systems where you have sort of populations of Daphnia. We've talked about trying to do that in the lab. We haven't really worked out the details about how to do that, but I mean, there's other possible ways to do these kinds of experiments.
certainly sort of like it motivated lots of possible hypotheses.
Yeah.
Like maybe even including the sort of evolutionary sort of like mechanisms maybe potentially. But then you say you only have 2 weeks, right? So so you just keep counting the same group of Daphnia, and then how many arrive and how many dead at that time.
Yeah.
And then when you introduce these animals, you have the same age, sort of the
Mhm.
same.
They all start out at
Same size.
less than 1 day old. Yeah. But this sort of makes me think of one, so I don't want to cut off your question, or your question, but it does actually make me think of one thing, which is like, so here I've shown this sort of stoichiometry of animal toxicity, but we shouldn't ignore sort of like how the toxin interacts with the food as well. So there's sort of like a whole nother level of interactions that we sort of need to think about. So like with nanosilver, which we've been working in the lab, adding algae to the container makes the nanosilver less toxic, but it's because the algae takes it out of the water and then the Daphnia is exposed to less. So those are sort of like additional interactions that sort of could be layered on to this type of study.
I think it's really interesting, and I think it is really important because, we're appreciating more and more that nutrients are another form of pollution in a way. Maybe not for Daphnia in the case of phosphorus, but it seems like you've kind of convinced yourself that it's true that under phosphorus-rich conditions that they actually do worse under toxic conditions. So I guess two questions. One is, can you relate these results to LC50s? I wasn't quite sure, or other toxic endpoints. Like, did you bracket those values? And it'd be interesting to see where the toxic effects start to arise, at least relative to dose. So that was the first question. And the second one was, I could see how there's this paradox. If you ramp up growth, it could in some cases increase exposure. If they're increasing their food consumption, it's a dietary-borne contaminant, for example.
So for the first question, funny story was like the very first paper we published on this, we didn't have LC50s, and then the reviewer came back and said, well, this could be acceptable, but you'd have to include LC50s. It's like, well, that's not really the point of it, and I think James figured out how to calculate the LC50s for us, and there was maybe a bit wider confidence intervals associated with that than what we'd want. And it's because the way that you calculate the LC50s. But I think your question is actually really interesting, which is there a way to sort of calculate where the toxic effects start appearing across this gradient? And we need a different experimental design to do that, but I think that's a great question. So sort of like, where does toxicity start to manifest itself, and does that change with food quality? So that's a great question, something that need a different experimental design, but we should do that.
Well, what I remember too is the LC, you might calculate that by saying, what's the LC5? And what I recall is those error bars are humongous. I mean, it's better for an LC50 than it is for an LC5, if I remember right. Is that somebody probably works at that kind of model all the time, more than I do now, but my recollection is those models aren't as good at calculating that as they are calculating the LC50.
I mean, the other problem with this is that when you get into the world of toxicology, it turns out that most data that you collect isn't very interesting, because either you expose them to a low concentration and everybody lives, or you expose them to a really high concentration, everybody dies. But that data is just as valuable as what's in the middle, but it seems that you're not, right? So we could repeat this experiment with a lot more concentrations to get at your question, but you'd have a lot of points here and it wouldn't seem very interesting. But without that, then we can't answer that kind of question. So you sort of have to accept this, that you're going to have a lot of what looks to be non-informative data, but it actually is informative.
Well, there's also, at those really high doses, if only 1% of the natural systems ever see those concentrations, what's the value of that versus doing something that's, 50% lower or much lower?
No, absolutely. But if we had repeated this experiment with only the environmentally relevant concentrations, we probably would have got a lot different reviewer comments, as we've seen in the past. So it's true.
I've got a question as it relates. I'm sorry. If it's a follow-up, go ahead. Let's follow up to the fellow from Liverpool. So my question isn't that long anyway, and some of it's been sort of brought up already, but just to sort of connect this to what's going on in ecotoxicology right now, is there is a lot of discussion about exposure routes and how important dietary exposure is as opposed to dissolved exposure. And I don't know necessarily how this answers this or helps address it, but I'm sure there's something in here that's, there's probably differences in the Daphnia exposure relative to their growth rate and the food quality that they're getting. So you may be getting, and you also made the point that there's also differences in the food, in the algae itself, and the where the contaminant is, whether it's attached to food particles or dissolved in the water. But this is an open question that's being sort of tackled by ecotoxicologists of, how important is dietary exposure relative to dissolved exposure, and thinking about partitioning of contaminants within these different exposure routes. So I think that might be a way that stoichiometry and the way people think about food sources and how much they're eating, what they're eating, could connect to ecotoxicology.
There's not a question, though.
No.
No.
Well, since I have the microphone, I guess what I would say is that I like the idea of asking a question related to a substance, okay, that is primarily dietary, but there are thousands of organic molecules that are, the relative exposure just flies across the gill relative to diet. So I think what you'd want to do is kind of like we were talking about the heuristic modeling approach, and then see what differs. I think that would be an example. But I guess my question for the group was really stimulated by the fellow, and I forgot your name, I'm sorry, from Liverpool. Have you been involved? Are you familiar with experiments that have actively manipulated N/P ratios, for example, and examined how that would affect bioaccumulation dynamics of inorganic or organic molecules?
Not directly. I know Mark Vine in Birmingham, he's doing similar things, but it's mostly to do with change of the media instead of food levels, which they see kind of similar effects. Depends on the media, what you put in, etc., what is available, how the compound is being made available to the daphnates. But specifically, CP ratios or NP ratios, no, I have not seen data like that yet.
Yeah, because the question would really, I think, be related to if organisms have varied internal CNP quotas, for example, would they be more or less likely to accumulate a certain type of molecule that could be related to lipids or whatever it happens to be. But I'm wondering what kind of work has actually been done in that space, because I'm not thinking of much right now.
Comment before was made about this being like a nutrient-rich like food source, basically. And my question are the ratios you're looking at, are they nutrient-rich or poor? Like, are we on the upper end of things that when there's the high carbon ratios that these are very much more on the nutrient-poor end of things, or is it compared to what would be available to them in the environment? So the assumption was made that this is a nutrient polluted or a nutrient enriched system, where I'm just wondering where baseline fits into these levels, basically.
Yeah, so I mentioned this at the beginning. CDP ratio of 100 to 300 is very typical in lakes. So the average, like global average, is like 250.
Okay.
But in the hundreds would be very normal.
Okay.
In some systems, CDP ratios in algae have been seen as high as 2,000, but there's only like a couple papers, and it was seen like a couple times. So a phosphorus poor lake might have CDP ratios of like 400 to 600 would be more typical.
So what we're exploring here is definitely more on the not in an over-enriched system. These are depleted systems, basically.
Well, CDP ratio 700 would be what you would see in like the most phosphorus poor lake.
Okay.
Whereas the 100 to 300 would be more typical of what you'd see in average lakes.
Okay.
And nutrient pollution would drive those ratios down. So in a very phosphorus-rich lake, you'd see a very high phosphorus content, so you'd see a lower CDP ratio.
Okay.
So that was one thought of mine, was like sort of linking it to global change, is if you're adding a lot of nutrients to these ecosystems, it's sort of like priming the systems for organisms to grow faster. This could increase the toxicity that you're seeing from other chemical stressors.
Yeah, I
We'll
just
hand
wanted
off.
to follow up on Brian's question. So I've done some work manipulating nitrogen to phosphorus ratios and looking at methylmercury bioaccumulation in Daphnia. And what I found was that with higher phosphorus diets, you get lower methylmercury bioaccumulation. So methylmercury assimilation efficiency and loss from excretion stays the same regardless of food quality. But what changes is just the weight gain in the Daphnia. So it really is pure growth dilution. So the methylmercury kind of biochemistry stays the same. At least that's what this indicates. But because there's so much more weight gain relative to the methylmercury gain, you get a really strong effect where you just get a much lower mass-specific methylmercury body burden on high phosphorus diets. And so that we found in the lab, and in field studies in the US, they've shown correlations between N to P ratios in lakes throughout the US and methylmercury concentrations in fish. However, I know that there are some new studies by another person named Roxanne, actually, who studies mercury as well. And she did some work in China, and I believe that she found that in even higher nutrient concentrations or phosphorus concentrations that you don't see that dilution effect. So we don't really know, but my suspicion is that you might see some kind of nonlinear effect. I suspect that in other lakes where there's super high phosphorus concentrations, that there might be something else going on. So I don't know of any other, but that's one example.
We're going to talk about a model shortly this afternoon that was mostly motivated by that data set you're talking about. Yeah. And then Greg Mayer at TTU and I have been just starting to collect data that's manipulating CNP in algae and Daphnia, and also keeping track of cadmium uptake. And which other ones are we looking at?
So we're doing a few metals. So we're doing trophic transfer. So we're looking at uptake in algae. We're looking at uptake in Daphnia. We're looking at uptake from the food source from the algae to the Daphnia, and with altered N to P ratios, C to P ratios, C to N ratios. And we're doing that with cadmium, with copper, and with arsenic. And the interesting thing, and not only am I homeless, by the way, I'm also labless. I've had 6,000 gallons of water spill into my lab 2 weeks ago, and again today I just got a call. So we're not doing it right now, per se. Right, we were. And what's really interesting, as opposed to the mercury stuff, is with arsenic, because arsenic, if you look at it, it's just a big phosphorus molecule, but with some extra electron shells, right? So it's kind of a mimic for that. So one of the other things we're doing is looking at phosphate transporters and upregulation, downregulation of transport of phosphate, or the mechanisms whereby to do that, and trying to figure out, well, if these are very phosphorus limited and they're upregulating phosphorus uptake transporters to try to get more phosphorus into the cell, are they also taking up more arsenic? Is that resultant of physiological change in the organism that's allowing them to take up more of the toxicant as well? And then what does that do to these growth curves? So that might be a little bit different than say, a methylmercury that doesn't really have a transporter that's related to the phosphorus or the nitrogen as much as it is to lipid bioaccumulation or something like that.
Yeah, so couple things. I always say that, I guess. Methylmercury is very much an end member, kind of an extreme example. So I think looking at the mechanisms like you're doing is really valuable in the way that we should go. I just wanted to point out, and I think it's great too that you're looking at that data set, and that was kind of some inspiration. But I just want to kind of toot my own horn a little bit further by pointing out a paper, only because it's relevant to this discussion, is that after finding that out about methylmercury, we did some modeling looking at all these different metals, just metals, and seeing what the potential is for growth dilution of different elements. And just if you only consider basically assimilation efficiency of the individual elements and efflux or loss rates of those, you can predict how important growth dilution is going to be. And it's most important for things like methylmercury that have very high assimilation efficiency. So you keep in most of what you eat and you lose very little over time. But for a lot of elements, zinc, copper, arsenic, pretty much everything else, because the loss rates essentially are lower for that it's much less sensitive. Body burdens are much less sensitive to growth dilutions in most cases of realistic, growth conditions.
Right.
So just without even thinking mechanistically about it, that was a prediction in an AMNAP paper that it'd be, if you're looking across different elements, you could see if it bears out.
Yeah, that's interesting.
Yeah.
I want to share my experience with toxicants and stoichiometry, because I did with my PhD student 2 experiments with gammarids, so organisms that grow during a molt cycle as about 20 days duration. And we did 2 kind of experiments. The first one, we fed organisms with different kind of resources, good quality and low quality resources, and only after 15 days we add the toxicant. And when we do that, we have results that are in accordance with your hypothesis. But when you do the contrary, when you do an experiment with different resource quality like you did, and with an exposure to the contaminant, to cadmium, exactly like what we did, we observe quite results that are quite in accordance with what you found. So our explanation is that when you add a good quality resource, you increase the molting frequency. And so we think that the molting event is the most sensitive period for the organism. So one question we have is it the same for organisms that are molting and non-molting? Because perhaps this is a molting event that is the most critical period for this organism. And in fact, when we look at organisms that died, all of them were died during the molting event. So it's just an observation, but perhaps it's something that happens. Because when they are molting, they intake water to grow and then make the cuticle. So perhaps it's a very critical stage for these organisms. And just a last comment on this result, we also had a lower growth rate. So we imagine that this is an apparent growth rate effect. So organisms that had the fastest growth rates died. And we, in fact, the growth rate that we measure is the growth rate of the remaining organisms. So the growth rate of organisms that had the lowest molting frequency.
Yeah,